Good morning, everyone. It's our great pleasure to welcome Vincent Debat to give the seminar today. So just a brief overview of Vincent's career. Vincent did actually his PhD in Montpellier, but he was a paleontologist then, working on the shape of things, rodents. Then he went to do quite a few postdoc projects in Gif-sur-Yvette to work with Jean David on Drosophila, and then he went to Riverside to work with Derek Roth, where he delved into quantitative genetics. And then he came back to Europe, to Manchester, to work with Chris Klingenberg, so a great morphometrician. And since then, he's been working at the Natural History Museum in Paris, working first on Drosophila, fluctuating asymmetry, and then but he's now actually the curator of the Lepidoptera butterfly collection in at the Natural History Museum, which is a great promotion, obviously. So Vincent's been working since then on butterflies and doing all sorts of interesting things with morphometrics and the shape of butterfly wings, and now flight, I suppose. Morphometrics on Morpho butterflies. Good. The floor is yours.
Thank you very much, Mathieu, for the introduction. So before I start, a few things. So I'm very happy to be here today. As Mathieu said, so I did my PhD in Montpellier, so I'm pretty glad to be back and to see some well-known faces. So it's my pleasure to be here. Second reason why I'm glad is because of the project I will be talking about. So I actually came to biology because of butterflies. As a child, I loved butterflies and I was collecting them. And so during my studies, I started working on different things. But now, as Mathieu said, I'm back on butterflies. And for a butterfly lover, working on Morpho butterflies is a bit like a dream project. So I'm super happy to be working with these guys and to give you an overview of what we've been doing in the past 10 days or so, 10 years. Sorry. Yeah, one disclaimer is that I'm really a phenotype person. So there won't be any genomics representation. Maybe one slide, but mostly a phenotype. Another important thing is that I've been leading this project in tight collaboration with in Collège de France in Paris. So I just want to acknowledge this right away at the beginning of my talk. And final thing before I start about sorry, this movie. So it's not 900 on the movie. It's unfortunately it was just 006. And the density, we were doing some macro capture, and it was quite low. So I was over optimistic using 3 digits for the numbering, but I will talk about that. Okay. So, starting with this picture of an Amazonian understory. So you all know when you see on that picture that kind of habitat is really packed with a huge diversity of species that tightly interact. And so ecological communities are strongly impacted by the evolutionary relationship between species that live in sympatry. So in our team, we are particularly interested in interaction between closely related species living in sympatry and how do these species coexist. And a related question is how these interaction shape the phenotypic evolution and vice versa. How does the phenotypic evolution of traits impact the interaction between species, the diversification, possibly speciation? So to investigate these questions, we focused on that model. So I made this composite picture using different individual images from each of the 30 morpho species. So this gives you an idea of the phenotypic diversity of this group. So why using morpho butterflies to do this research? So there are different reasons. So personally, I really love working on this because they're amazing animals, but that's not obviously the main reason. So one really important reason is that we are talking about butterflies that are tightly related within the same genus. And there is a huge phenotypic diversity. You can see that although morphos are super famous because of the blue iridescence of the dorsal side of the wings in some species, you can see on this picture that not all morpho species are blue. There's huge variation in color pattern and coloration itself. Even if you look at the blue morphos, there's a lot of variation in terms of amount of blue on the wing, but also the blue itself. So the blue coloration is really strongly variable. That's one aspect of the phenotype. But so there's a scale bar at the bottom. You see that morpho, I didn't say that, but morpho are only neotropical bugs. They only live in Central and South America. And compared to all European standards, they are really, really large butterflies. So the smallest species is this one, but you've got really giant species that reach almost 20 centimeters in wingspan, almost among the largest butterflies on Earth. So huge diversity in terms of body size. And you can also see on this slide that not only size varies, but also shape. Huge diversity of wing shape. For example, this one has really elongated forewings, while you have other species that show some more compact wings. I don't know if online they see my the mouth. I can show on the screen. Yeah. Okay. So some species that have quite rounded wing shapes, and some other pointy and with tiny tails on the hind leg. So the question is, what are the evolutionary drivers of this huge phenotypic diversity? Another interesting thing is that morpho display quite a lot of a high degree of sympatry. We can find up to 12 morpho species living in the very same localities. And if you stay at some place for long enough, you will see these 12 species flying around. So what are the interactions of these species, and how does this relate to phenotypic evolution? We started investigating wing shape, and I initiated this project with Marianne Elias and during the PhD thesis of Nicolas Chazot. And working at the museum is quite nice because our lab is at the first floor. And climbing just one stair, we access to the collections. And in Paris, we have the largest public collections of Morpho in the world. So to sample, we just need to climb one stair. So we open the drawers and start sampling the different species of Morpho. First thing we did was that Nicolas did was updating the phylogenies to get historical context to this study. So he sequenced a bunch of genes, and he got this phylogenetic structure, which is now well established, well stabilized. So we've got these two species, Morpho that branch at the basis, and then we've got two big clades, one with 20 species, and this one with 10 species. I will go back to that. Okay, so when I started working on butterflies almost 10 years ago, I was expecting that we knew many things about their evolution and their ecology, just because they are among the most famous insects ever. They are found in every curiosity cabinet. They are really famous. But I realized that we don't know much. They've been super heavily collected and sold, but nobody has ever really taken enough time to investigate the ecology. So we don't know much, but one thing that was quite clear was that you can roughly assign the different species to either canopy species or understory species, just because when you're in the field, you can see some of the species flying at your level, and for some others, you really need to look up and you see them really high in the canopy. So this microhabitat partition roughly makes sense. And so we wondered whether the evolution of wing shape might be affected by this microhabitat structure. Because, I mean, you can see on this picture, but the understory in the Amazonia is really a cluttered environment, really dense. And for butterflies of that kind of size, it's not the same as flying in the open canopy. So various other things vary with the height. It's not the same community of predators flying at the top of the trees and in the understory. So various parameters that might impact the evolution of the shape. So we need, and this is, so the color code I'm using is green for understory species and blue for the canopy species. And unfortunately, it matches perfectly with the phylogeny. So this reduces the power we have to detect some impact of the microhabitat, but that's the way it is. So we've got this clade that has shifted towards the canopy. We can think that the ancestral state was more the understory, flying in the understory, because these two species branch there, and the outgroups are all understory species. So likely the flying in the canopy is a secondary evolution. So we did some morphometrics on this. And when I was doing my PhD with Jean-Christophe at ISYEB, I remember that I was joking that we should do the la Morpho des Morphos. And basically that's what I did with this project. So I will go quite fast on that. So we defined some landmarks and semi-landmarks on the wings to precisely describe the wing shape of both the forewing and the hindwing. Then we superimpose the different configuration to get rid of all the differences that are not due to differences in shape. We put them at the same scale, we place them in the same place of the plan, and then we rotate the configuration so that to optimize superimposition of the landmark. And then we work on the superimposed coordinates as shape variables in multivariate statistics. So that's typically the kind of thing we do, principal component analysis. And on this one, I'm only showing the species mean. It's an analysis that is run on the full data set, almost 900 individuals, but I'm just showing the means. And the closer to point in the morphospace, the more similar the wing shape, the corresponding wing shapes. So you see that there's super strong microhabitat effect along the PC2. So a nice thing with morphometrics, geometric morphometrics, is that we can visualize the shape changes associated. So you can see that along the PC2, the canopy species tend to have broad wing shapes with some tiny indentation or tiny tails, and more contracted wing shapes for the understory species. So we can project the phylogeny on the PC plane and assess the phylogenetic signals, which is super strong in this case, which is obvious, strong phylogenetic clustering. But still, we could apply some phylogenetic tests to assess the divergence between microhabitat. And although the ecological partitioning matches the phylogeny, the divergence is stronger than expected in a neutral, I think, Brownian model of evolution. So this points at an adaptive divergence between the two microhabitats. So that is a way to combine the information of the two different wings, and you see that the covariation is really strongly driven by the opposition between canopy and understory species. And it opposes the triangular pointy wings of the canopy species to shorter, more rounded wing shapes in the understory. And this makes sense in terms of ecology, because, I mean, if you talk to people doing aerodynamics, having long, pointy wings might be associated with improved performance of gliding flight, which might be advantageous to fly in the open canopy where there are quite a lot of wind and things like that. While flying in the understory, where it's really cluttered, you might think that maneuverable and strong flapping flight might be favored. So to test that, we needed to go on the field and then to start investigating the evolution of flight of the plane. So this was the PhD project of Camilo Waz, super talented grad student. And so we went to North Peru in one of these spots where there's a huge diversity of morphos in particular, and we started collecting and filming the morpho. So we first spent a lot of time filming the natural behavior of wild morpho to get an idea of the natural behavior before switching to captivity and fine quantification. That's an example of the flight of a canopy species. So you see that this one glides quite a lot. And this is really remarkable because, I mean, almost no insects actually glide. Butterflies are well known for their gliding abilities. They do what's called flat gliding flight. But that kind of long gliding phases is really unique in butterflies. Really long gliding phases. So that's another movie we took. This time it was in French Guiana, so we're using the drone. So you see that they almost don't flap at all for really long periods of time, which is really, really, really cool. And it's really nice to see. So an example of an understory species. So you see, it's really a different type of flight. It's really flapping flight. The trajectory is really, really erratic, how it's sort of bouncing flight. And there are also some visual effects of. I will go back to the color later. So we built a cage on the field, really nearby the forest where this guy fly, and we installed a 3D stereographic video system. Actually, we installed 3 GoPros, and we brought back the butterfly that we released in the cage for flight quantification. So that's the kind of things that we did. So the captivity did not, I mean, quite impacted the flight behavior, but we could get some fairly natural flight sequences. I mean, we did some quantitative comparison between the two conditions in the wild and in the cage, and it was satisfying. So doing that, it's possible then to subtract the background, and then using some deep learning algorithms, really nice tracking devices now. It's possible to reconstruct then the 3D trajectory by combining the different views, and then to extract the flight parameters. So basic stuff, upstroke, downstroke, quantifying the relative importance of gliding and flapping phases. But obviously also more precise quantitative parameters like flight speed, acceleration, angular acceleration, and some more precise parameters. And we analyze all these parameters together as a multivariate analysis with like what we do with morphometrics. And we get that. So the PCA, each dot is one single flight trajectory and the same color code, blue canopy, green understory. It's a fairly strong divergence in terms of flight behavior, flight trajectories between the two. Again, using phylogenetic tests, this divergence goes beyond the neutral expectation, so pointing at possibly adaptive divergence of flight between these two. And if you look at the PCs, in PC1, it's a common variation in terms of the amount of gliding flights between the two, which might be a bit surprising, but what we found was in captivity, they didn't glide at all. So really, not at all, but it was really strongly reduced. So yeah, we are not picking this variation. So the second axis that separates the two groups mostly involves the flight power, with understory species at the top flying way faster with more powerful, more strong acceleration, and while the canopy species tend to fly more slowly and more erratic trajectory. So still considering the gliding flights, I said they still glide a bit, and we focused on that parameter, which is the gliding angle, which is shown on this on the right part. So an efficient gliding flight allows you to go far while losing a minimal amount of height. So a good glider should have a shallow gliding angle, which is the case for the canopy species. So this suggests that these guys can glide way better than the understory species, and that matches the observation in the wild. So then we ask whether this difference in gliding efficiency is actually due to the differences in wing shape that we quantified. So to test that, we started collaborating with physicists in the Netherlands, and with Paul Muséum in particular, doing some aerodynamic simulation of fluid dynamics. So we used the movies that we took in the cage to get the precise wing position during the gliding phases, and as a starting base to build a 3D model that was used in the aerodynamic simulation. So actually what these guys do is that they simulate locally on each point on the wing the airflow, and then they integrate all these local simulation around the whole wing shape to assess the airflow around the butterfly while gliding. And it's possible to change the shape as input of this model, and to change also the position, that is the angle of attack of the shapes. And so this is roughly, I mean, the main result of this analysis. So on the right part are 3 canopy species wing shape. On the left are 3 understory wing shapes. So it's quite complicated, but the important thing is these purple things in the middle that you see only on the understory wing shapes. And these are vortices that are produced while gliding. And this imposes a huge amount of drag to the butterfly, sort of slows down the butterfly while gliding. And this doesn't happen in the canopy wing shapes. And so these parameters lift to drag ratio that directly estimates the gliding ability. And as you can see, it's always the same at every position of the wings. You've got a higher efficiency of gliding flight in the canopy species. So better gliding performances in canopy species. So if we combine all these, so the visual observation and quantification in the wild, the 3D kinematics, there is a tracking of the 3D trajectories in the cage and the CFD simulations, all compared to point at an adaptive divergence of flight between understory and canopy. So if I go back to the question, so the coexistence in sympatry, might be favored by this vertical stratification of the habitat, the splitting of the space between the two type of the two group of species that goes together with a divergence of flight and divergence of wing shape. So we don't know whether what is the precise cause of this divergence, whether there were some competition among species in the understory that might have favored the vertical shift or not. And what kind of competition? So one possibility is sexual interference. I will go back to that later. Another thing that we don't know is the sequence of this phenotypic evolution, whether wing shape facilitated the colonization of canopy, or whether it was a secondary thing and first a shift in behavior. That is something we don't know. Another thing which is interesting, whether speciation was involved at all in this process, whether there was some sympatric speciation splitting the two groups, or whether speciation occurred in allopatry, and then the species came back into contact and then shifted in vertical strata. And then we might also, some reason point out some really strong consequences on the evolution of coloration. And that's what I'm going to present now. Okay, so this particular part of the project was mostly based on the PhD thesis of Agathe Puissant that just defended into 2024, and Joséphine Ledamoiselle that is still working on that. Just a small movie to show you it was a Morpho iridescence look like, so really, really strong visual effect. So if we go back to the global image of these, you see that there's strong variation in iridescence. This is a structuring in terms of vertical strata. The three columns on the right are the understory species, and the two on the left are canopy species. So it doesn't really match with the opposition in color. You've got three species in the canopy that are blue, so most of these ones are blue. But as a matter of fact, one thing that we got is that all the non-blue species are the ones that actually glide. And the other, in particular, these three canopy species, so we quantified the flight of this one. It doesn't, it's really puzzling because it has really elongating wing shapes, and the aerodynamic models predict that it's super strong glider. And actually, when you see it in the wild, it doesn't glide much. It does some really short and really fast glides, but it's really not a glider. This one, we saw it in Panama, and we didn't quantify, but definitely not a glider. And this one only flies in the Atlantic forest in Brazil. So I was lucky enough to go there last month. It was really an interesting species because it's blue and it's iridescent, but it's a matte variation. It's not a bright and flashing species, so it's really peculiar. We were wondering whether this one would be gliding or not, and it doesn't glide at all. So there's really a nice match between the loss of blue iridescent and the evolution of gliding flight. So this might give a hint on the selective pressures acting on the evolution of the blue iridescent. And we think that the reason why there is this connection between flight and the evolution of blue color is due to the dorsal-ventral contrast. So this is the same individuals that I just flipped, and I'm showing the ventral side. And you see that all Morpho are brownish on the ventral side. So strong dorsal-ventral contrast. And what does this do? That you don't see super well, but you see some blue flashes. And so I'm a human predator of Morphos. And it's really easy to see Morpho from far, easy to spot them. But as soon as you get closer from them, because they fly super fast and a really erratic trajectory, it's really difficult to catch them. And if you miss the first attempt, then no way, they stop flying all over, and it's impossible to catch. So we think that this might be happening with natural predators. And there are data suggesting that insectivorous birds, they tend not to attack morphos when morpho are passive. And when they're trying to catch them, most of the time they fail to catch them. And possibly, you might also associate blue coloration with this major difficulty of capture. So what we think at the moment is that the dorsal blue clearly enhances the difficulty of capture, but in turn, it might be used by birds to identify this difficulty of capture. So it might signal the difficulty of capture. So this might tell remind you of something which is really famous in butterfly. What happens with electronic butterflies? You know that these are toxic butterflies and really aposematic, or really intensely colored. And birds learn to associate the bad taste of toxicity with this bright coloration. And what does it do in terms of macroevolution? It leads to the convergence of local toxic species, so Mullerian mimicry. What's happening in morphos? Birds might associate the blue coloration with the major difficulty of capture. So this might be the same as toxicity, but difficulty of capture instead. This might induce what is called escape mimicry, so local convergence of different fast-flying species around the same phenotype that is known and avoided by predators. So we tested this hypothesis focusing on a triplet of species that fly in the Amazonia. So Morpho Eleanor first is, so that's the most widespread species of Morpho. You find it until Mexico and to the north of Argentina. And it's really variable geographically. And in particular, what is variable is the width of the blue band. You see the one in French Guiana has really small blue band, while in some other places, and even other places in Central America, and they're almost completely blue. So lots of geographic variation in the blue band. And so there are two other species, Morpho Achilles, which is actually the sister species of Morpho Eleanor, and another a bit more distantly related species, Morpho Deidamia. And these two species are always in sympatry with Morpho Eleanor, just because Morpho Eleanor has a larger distribution range. And what is striking is that the two other species, they display seemingly from a shallow look at the collections, really parallel geographic variation with Morpho Eleanor. So what we did is that we quantify this parallelism of the local convergence. So this is the situation, the two sister species and more distantly. So we went up to the collections and we sampled. We had a big enough collection to get intraspecific variability. So really nice sampling. And we sampled both populations where Eleanor is just alone without the two other species, and many population of the common distribution range. We use some the color pattern modeling, so it's a method to quantify the color pattern developed by a former student of Mathieu, Yann Lepou, some of you might know him, to precisely quantify the color pattern. So I'm not going into the details, but it allows finally quantifying and visualizing the differences in color patterns, and this is a PCA on the color pattern data. So we've got three different type of symbols, and the different colors are for different localities. And what is pretty clear is that the different colors, they tend to cluster, which means that the different species tend to be in the same part of the morphospace, irrespective to the specific stages. But they converge when they are in sympatry. So we tested that doing some generating a null distribution, comparing species by pair, and just randomly picking one of these two species and computing a morphological distance between the two. So that's the distribution you got here. And we compared it to the real distance in sympatry. And in sympatry, most of the distances are way smaller than the expected distance if we don't take the sympatry into account. So this really shows that there's a tight convergence in the color pattern. Okay, so this is the color pattern, but I mentioned iridescence. So iridescence is the variation of color of the light reflectance, depending on the angles of illumination, but also depending on the angle of observation. So that's something that is super difficult to measure. I will go back to that just after. But so we asked whether this convergence in color pattern extended to iridescence itself, because in many examples of structural coloration, it's been suggested that these colorations are often involved in species recognition or in mate choice. And so if this is the case, we would rather expect that the sympatric species would diverge in sympatry if individuals use iridescence to identify their own species from the other. So we went for testing that. So the aim is to quantify iridescence in the very same population that for which we quantify color patterns. So we haven't done that. It's really time consuming. So I'm just showing you two different situations. So first, the sympatry between Eleanor and Achilles in French Guiana, and another situation comparing two allopatric populations, but within the species Morpho Eleanor. And contrasting these two situations might give us a hint on whether it's converging, iridescence converging in sympatry or not. But as I said, measuring iridescence is really tricky. So we place the wings, and then we work with two fibers, so with a spectral photometer, and we vary the angle of lighting. And at each of these positions, we need to vary the angle of observation. So we have many combinations of these two angles. So having a really precise quantification globally of iridescence is really impossible. It's really way super strongly multidimensional. So we focus only on a few positions, actually 21 positions. And the kind of data we got is that, so a reflectance spectra across the different light wavelengths. So these can vary in the height of the, so this is a variation in brightness. So very bright butterfly will reflect a lot of light. So this shows in the height of the curve. And different colors in the everyday sense would involve a shift, horizontal shift of the main peak. So this is called U. And so it's possible to quantify iridescence by focusing on a few synthetic parameters, which is U, brightness, and chroma. Chroma is the purity of the color, which is the width of the peak. So there are actually two different ways to quantify iridescence. Either you consider the all different spectra that you measured at all different angles, and you put that all together in this big multivariate data set, and that you analyze using multivariate statistics, a bit like I presented before. Or you can focus on this summary statistic, U, brightness, chroma, and just depict the angular variation of these parameters. So that's what we do. So first, I'm showing you the two populations of Eleanor that are in allopath. So this is a PCA using the whole data set. So each dot is a summary of all the iridescence variation. So the closer 2 points, the more similar the iridescence. So you can see on top that there's a difference, a significant difference in iridescence between the two subspecies. So being on the two sides of the Andes actually makes a difference within species. They don't have the same iridescence, and also a difference in sex. The males and females do not have the same iridescence. So if we look at the specific variation in U, you see that roughly the shape of the sort of reaction norm are roughly the same, but they do not have the same elevation. So which means that the color itself is not really varying in a different way, but the colors are different between the different groups. And to interpret this in ecological terms, we use some visual models to tell whether predators or butterflies would perceive this difference. And actually being above this threshold suggests that these differences can be perceived both by butterflies themselves or by predators. We did the same for the sympatric species, and this time the species effect is no longer significant. There's a sexual dimorphism, but no longer different, and the species are not different in terms of iridescence. The same for you, really similar color and same variation between the two, which means that the iridescence is indeed converging between these sympatric species. And if we look at the visual model, they are below the threshold. That means that the subtle differences cannot be perceived either by predators or by butterflies themselves. So this shows that clearly iridescence is also convergent in sympatry. So if we think of what might be the selective drivers of the evolution of the iridescence, this points towards an effect of selection by predators that might favor the local convergence of iridescence rather than an effect related to species identification or mass choice. Okay, so it's likely the coexistence within the understory might have promoted the evolution of escape mimicry. And an interesting problem is that we are talking about in Morpho Eleanor and Achilles, they are sister species. So sister species, they are diverged since 2 million years ago. They are in sympatry. They are super convergent. So this calls of the question of how do they maintain isolation and whether there might be some reproductive interference. So I'm presenting, I think that's the last experiment that I'm presenting. So back to Peru, we built a solar powered device in which we pasted the wings of the real Morpho, sort of mimic the flight of a Morpho in the wild. So we first rinsed the wings with hexane that allows to remove all the possible pheromones and to be sure that the only signals that might be acting are visual. And so we put that on the riverbank with the 4 sticks to delimit an interaction space to sort of code the behavior of wild Morphos, and also add some cameras to record the interaction. So we coded the different behavior this way. So if a Morpho was passing in the background but not interested, so that was passing. In case where there was a marked deflection in the path, in the trajectory, but that the Morpho was not entering the space, it was just an approach. And in quite a lot of cases, we have that kind of thing. So really tight interaction with the wild Morpho flying around. So we use the 2 cameras to track the trajectory both of the dummy butterfly and the visiting butterfly. And this was really important to finally quantify the behavior, the flight behavior of the visiting butterfly, because obviously what we did is that we changed the wings on the setup. So we used male wings and we also used female wings, and it was nice to have the precise behavior to tell whether they were behaving the same for males or female dummies, and obviously between the different species. So I don't have time to show you the precise result of that. But the conclusion of that experiment was there's really strong sexual interference. The males tend to fight with other males from different species. They tend to court the wrong females. This suggests that, yeah, they might just make some mistakes and spend some time wasting some time in interspecific interaction. So this might suggest that there might be some gene flow between the 2 species. We have some genomic data that says that there's no recent gene flow between species, which suggests that there might be some other forces at play that prevent this gene flow to occur. So we did a capture, mark, recapture work. We spent 3 weeks capturing every single Morpho that we could catch, marking them. And what we discovered then is that we noted the flight hour. And the different species of Morphos, they don't fly at the same hour. And in particular, Morpho Eleanor and Morpho Achilles, Eleanor fly around 11 in the morning, and Achilles fly after afternoon at 12 and 1 p.m. And this is fairly stable. If we plot the time of flight of the first capture and the recapture, it's fairly constant. So this divergence of flight activity time might indeed mitigate this reproductive interference, and in the wild they do not really encounter one another that often. And this might explain the lack of recent gene flow between the two. So we are really currently working quite a lot on that time divergence, because that's really an interesting thing. And Morpho are really amazing for that. There's one single species, so I don't have any data to show you, but that only flies in the morning between 6 and 6:25. And that means that when to go to sit, you need to wake up super early, go in the forest in pitch black with your headlight, and say, it's impossible. There won't be anything flying. And just when the sun starts to rise, when the lights become grayish, you start seeing some light blue flashes, and it stops at 6:30, it's gone, and we don't know at all what these guys are doing the rest of the time. Really funny things. But we think that, possibly there's some kind of competition in the understory that might favor the divergence in this time dimension of the. Okay, so I've got one genomic side. So we did some sequencing of different genomes. So that's Manuela Lopez Villavicencio that is doing that. And so we have 11 reference genomes for the 11 Morpho species. So we investigated the evolution of the genomes, and we found some inversions that are mostly concentrated on the Z chromosome or on these guys. And in relation to what I just mentioned, we looked at the genes that are involved in circadian activity, and we found that the gene period that is on the Z chromosome shows some signs of acceleration. Whether these are actually involved in the divergences in a flight time niches is interesting possibility. That's it with the genome. Okay, so escape mimicry might induce or is associated with the convergence in color pattern, but also seemingly of iridescence. This in turn induces some reproductive interference that might favor some divergence in the flight activity time. So to conclude, we have this divergence in flight height that might have induced the divergence in wing shape and flight, divergence in flight height, convergence in wing color pattern with the evolution of escape mimicry, that in turn induces some reproductive interference that might favor the divergence in flight time, with possibly some additional consequences on the evolution of all the phenotypic traits. For example, it's not at all the same to fly super early in the morning in terms of thermoregulation. We are exploring that, whether there's a divergence in the thermal tolerance in these guys. But it's also super different in terms of visual acuity and visual sensitivity. So that's one of the projects we are now developing. So we were lucky enough to get some major funding on this Morpho project. So this HFSP project that I'm leading. So we are keeping on working on the evolution of flight, and in particular, finally quantifying the escape flight behavior. And we are predicting that there's a tight coevolution between iridescence and the way these guys escape predators. So this is done in collaboration with our physicist colleagues. We want to finally quantify iridescence. So I've just showed you a quick and dirty way to estimate iridescence, where we need to quantify that way more precisely and at the whole genus level. So that's a job done by some physicist colleagues in Colombia. And we are also working quite a lot on the evolution of vision, because obviously Morpho might be particularly sensitive to blue wavelengths. They might be super good at telling apart some subtle differences in coloration. So the 5th part is the PhD thesis of Raphaël Dupillier that is actually currently filming some butterflies in Peru. And the vision part is a part of the PhD thesis of Joséphine Le Damoiselle. So she sequenced, and using the genome data, she looked at the sequence variation of opsin genes that are the photoreceptors. And so she found some duplication, 2 different duplications in Morphos, and with some amino acid substitution that are predicted to induce some shift towards a blue sensitivity, so somewhat matching the predictions. And we're also working with some electrophysiologists, people in Slovenia that do some nice experimental work on the visual sensitivity of Morphos depending on the different wavelengths. And Violaine got an ERC funding. So while the HFSP project is more based at the macro evolutionary scales, the ERC is more focused on the micro evolutionary scale. So she's studying in detail the interaction between Morpho Eleanor and Morpho Achilles and documenting the ecological factors at play, so working on reproductive interference prediction. And she also developed the genomic analysis using genomes of various populations. This will be really important to date the history of colonization of the different localities and to be able to know which species came first, whether they speciated locally or whether it was shifted in time or things like that. So this involves the work of Manuela and Ricardo, that's how you might know, that is a postdoc at Collège de France in Violaine, and Erika Paez, also a postdoc, and Tite Fondinet, PhD students. And with that, I'm done, and I thank you for your attention, and I thank my collaborators for the super nice work. And I'm happy to take any questions. Just a bit of ad. We made a movie with Violaine, so it's a 30-minute movie, and we're explaining all this project. So it's aimed at the general audience, but I really like the movie. And if you're interested, please have a look. I think they're just Googling Morpho Papillon one should make it. Thank you, Vincent. Do you have any questions?
Do you have info on predators' behavior? Because if blue allows the butterfly to not be catch easily, and some canopy species lost this color, you have any difference in predators?
So yeah, I didn't explain the loss of blue in the canopy, in the gliding canopy, but if you think if indeed the mechanism of dorsal-ventral contrast and the production of blue flashes is actually under selection by predators. If you start gliding mostly, so you become a super stable target, easy to catch. So that's the hypothesis that explain why the loss of blue is associated with the evolution of gliding behavior in the canopy. That's one thing. So there are published data on predators. There are 2 papers, so by Brazilian colleagues that spend a lot of time in the wild, just standing hidden in the forest with goggles to quantify or to try to quantify the behavior of bird predators. And so they recorded the number of butterflies passing by, the species, and the behavior of birds. And they concluded that, I mentioned that briefly, when it's a morpho flying, most of the time they don't attack. They're not interested. And when they do, most of the time they fail to catch the butterfly. This suggests that indeed this might be working. We want to quantify that more precisely. So there's a postdoc. We're not involved in this project, which is a postdoc in STRI in Panama, and her project is to quantify the behavior of birds in captivity, releasing some morphos, and for example, masking the blue. You would be expecting that if these guys are no longer blue, you don't have the flash effect anymore, and maybe the birds might be better at catching them. But it's super hard. I mean, in captivity, it's not working apparently. So we also did another thing with the postdoc of Erika Paez. So they did some experiments in Ecuador, and they used some white sheets with lights that are used actually by bird watchers, because birds, they know that when there's this light, there are plenty of insects coming at the sheet, and that it's easy to get some free food there. And so what they did is that they placed some Morpho early in the morning on these sheets to see whether the birds would be attacking them. But it's not natural condition, because at that moment of the day, in that part, there's no Morpho, and so it's really, and the results are not really convincing. It's really tricky thing to measure, but super important, because I mean, that's a selective pressure that we are hypothesis. Questions? One question online. But yeah, Lisa. No,
We will. Robert, you can ask your question. Can you hear us?
Yes, I can hear you. I don't know if you can hear me. So I was wondering about whether these subjects, so a male and a female of the different species, are still technically able to reproduce, because you showed that they are flying in different times, but there's quite some overlap, and they will meet certainly a lot of times still.
So whether they are actually isolated or not, or whether they can actually interbreed or.
Yeah, because you said there is no gene transfer between these species, so it seems that they are not doing it. But I mean, how they are differentiating themselves, or I don't know.
So that's an interesting question. So I didn't mention it, but we also starting to get some chemical information, so on the pheromones. That's an obvious possibility that these guys recognize each other by scent, and indeed, it seems that there's a divergence of chemical bouquet in sympatry that is strong. I mean, when comparing the two situations that I mentioned for the iridescence, there's more divergence in sympatry than in allopatry between different populations of Eleanor and Achilles. So it suggests that they might be using pheromones to in addition to the time of flight.
Okay, thank you.
Yeah, thanks for the nice talk. You were interpreting the convergence in sympatry as something like Mullerian mimicry, which would mean that they basically adapt to each other to look the same. But I wonder in this case if the simple explanation is just that they converge to adapt to the same external environment, because contrary to Mullerian mimicry, where the signal doesn't seem to have any functional effect, here it seems to affect their performance in terms of confusing the predator and things. So an alternative is just like there's higher predatory pressure and then they convert to this thing. So could you test this?
So we wondered about that, whether the geographic variation could be somehow adaptive. It's locally select for the same phenotype in different species.
Yeah.
But there's really nothing obvious. There's no difference in elevation that could be associated with difference in the amount of black on the wing, for example. And really, we don't see any ecological, not anything nothing obvious.
Even the type of predator.
The alternative is more like geographic divergence. It's just neutral. It could be whatever color. But as soon as local predators start to recognize that and to avoid that, so this imposes some new selection on the other species. So it all depends on who came first locally and who is the most abundant.
I was wondering how you really managed to measure the convergence for iridescence in the sense that, if I understand correctly, they live at different periods of the day. And so probably the angle of the light differ according to when they actually fly. And so you really take into account what the time of the day, so the angle, to see whether the convergence really happen, taking into account the light condition that is actually occurring?
So no, we haven't done that at the moment. So in the vision model we assume it's standard light condition, and that's what is always done in the literature. So actually, you're perfectly right that if light condition actually changes during the day, we might need to change that. So we haven't done it so far. But we have, so I mentioned this colleague, Gregor Beluzic, that is really an expert in vision electrophysiology. And what he's done is exactly that. So I'm not expecting that between 11 and 9:30 light condition would change. I think it's roughly the same and so on. But this would be actually acting for these two species that we are, and it might be worth looking. But what he did, measured iridescence of the super early species. So clearly, light conditions are completely different at that moment. And he measured the iridescence of another later species. And he brought in the field in French Guiana, he brought some spectrophotometer, the environmental spectrophotometer, finally quantifies the light condition at these two moments. And when you combine the two, you get the color spectra in the very specific light conditions, they are identical. So he's super excited. He's saying that, okay, it's sensory drive that is responsible for the evolution of iridescence in Morpho. So we need to precise that, but that's super intriguing possibility. Yeah.
Following on that, there's probably other environmental factors that could affect the amount of light that goes into the forest, like the structure of the forest. So the latitude, for instance, that's maybe something that can explain the variation between population.
I'm not expecting really that light conditions are varying that much geographically. It might be worth really finally quantifying the variation in light conditions. But I mean, there's so much variation during the day. So we might need to measure that more precisely. But the habitat has really an impact on polarization, for example, light polarization. So we are also investigating that. So we don't know at the moment whether they are somewhat sensitive to a light polarization, but whether they are differently sensitive, that's another thing to measure. But we are actually investigating that possibility, but I don't know for the moment. Yeah.
Just wondered what the butterflies are doing during the day, if they are just sleeping away, not hunting for places to opposite or things like that. So,
I mean, I told you that we don't know much about the ecology of Morpho. We are starting to know better.
At least they fly several days.
But yeah, I know. They fly super long. In the capture, macro capture, the longer delay we got between two capture was 3 months. So we capture one specimen, brand new pristine specimen in early July, and we recapture it by the end of August, completely shredded.
You're basically waiting for resources to be available.
So seasonality and phenology, so that's really an important factor. And there are no precise data on the variation of the time of activity. I'm talking about year-long variation for morphos. But clearly, when we go in some places, we never really know what species will be flying at that point. So we have local colleagues, so we try to adjust. But sometimes we go to sample and there's nothing, and we need to go 1 month later and then there's a bloom. And so it's and people always tell us that everything is changing with climate change, and that El Nino is not the same, and La Nina, and that it's not reliable. It's not. I don't know, really know.
I was wondering, when you used the dummy specimens, did you have any predator interactions with the?
A good question. I don't think so. Yeah, no. But the thing that Camille was always around or to check the cameras. So yeah, it's super shy. So I don't think he saw any interaction with the dummy.
Okay, and completely unrelated, but with such intense blue coloration, do you know if the species can see ultraviolet lights and?
Oh yeah, they can. Or so the butterflies have ancestrally 3 different opsins: one in the UV, one in the blue, and one for long wavelengths. And so definitely they can see.
And have you ever tried to look at the patterns of the wings?
Yeah, we did some tries, but it's super difficult to measure on morphos just because the cameras are completely they saturate. And so you've got something that reflects super high, even when using just UV cameras, but it's not necessarily just because of specific. So it's difficult to measure. We need to do some. I'm not really good with that, but it's technically difficult with Morpho.
Okay.
So, there are some things. So if you look at the ventral surface, which is more matte, then you can see some UV reflectance in the eye spots. So they have eye spots on the ventral side that might also play an important ecological role, either in deflecting predators' attack away from the vital body parts, or also in sexual selection. So in some other species, it's been shown to be important. Okay, well, thank you very much, Vincent.